First, thank you for the kind presentation. And today I will present you the results of the project that I developed during the first year of my postdoctoral stay. And well, uh, we need, as an introduction, Alzheimer's disease is the most common form of dementia. The clinical diagnosis is characterized by impairments in memory, progressive cognitive dysfunction, alteration in mood and behavior, and presence of neuropsychiatric symptoms, and is frequent in aged persons. The clinical course reflects the underlying histopathological hallmarks such as amyloid beta plaques, neurofibrillary tangles composed by the hyperphosphorylated tau protein, neuronal cell death, and progressive neurodegeneration of neocortex and hippocampus. In this image, I show you the progression of the two main histopathological hallmarks of Alzheimer's disease. This progression is according to the model proposed by Brack and Brack, and in red, you can observe the A beta plaques, and in green, you can observe the neurofibrillary tangles. In the early stages of the disease, there is a little distribution of the neurofibrillary tangles in the entorhinal cortex and is absent in the hippocampus. In these stages, there's no evidence of clinical symptoms in the majority of patients. However, as the disease progresses, the distribution and quantity of plaques and tangles are increased. These stages may be silent, or some alteration can be appear, such as impairment in cognitive and mood disorders. Finally, in later stages, the amyloid plaques and tangles are distributed not only in hippocampus, also in neocortical areas, and these stages are characterized by an advanced dementia. And it's important to mention that one of the main structures damaged is the hippocampus. The hippocampus is a brain region that is presented in all mammals. This structure is composed by the CA1 and CA1 to CA4 and dentate gyrus. The hippocampus participates in a crucial manner in several functions, such as memory and learning, exploration behavior, regulation of stress response, and mood regulation. The hippocampus represents a bottleneck in processing the information. The first input to the classical hippocampal presynaptic circuit are the dentate granule cells of the dentate gyrus. These cells receive innervation from entorhinal cortex. In this regard, the morphology of dentate granule cells plays a significant role. Using Golgi-Cox staining, we can observe the morphology of dentate granule cells of different species. The common morphology of the dentate granule cells are The common morphology of dentate granule cells is composed by one primary apical dendrite that ramifies until form a dense dendritic tree. This morphology has been named Y-shape. It's important to mention that the morphological maturation of the dentate granule cells is crucial for the correct establishment of afferent connection from the entorhinal cortex. The dentate gyrus is composed by the molecular layer granule cell layer, and hilus. The outer granule cell layer, it refers to the area in which cells are in close contact with the molecular layer. On the other hand, the inner granule cell layer, it refers to the area in which cells are in close contact with the hilus. One distinctive characteristic of the dentate gyrus is the capacity to generate new dentate granule cells in the adulthood through a process called adult hippocampal neurogenesis. This phenomenon was discovered by Joseph Allman in the 60s in the rat brain, and based on this finding, several researchers have been carried out on this subject, demonstrating the continuous addition of dentine granule cells not only in rodent brain, also in the adult human brain. In this regard, it has been demonstrated in rodents that the position and morphology of the dentine granule cells in the granule cell layer vary according to the developmental origin. The developmentally born dentine granule cells are preferentially located in the outer granule cell layer, whereas adult-born dentine granule cells remain in the inner granule cell layer. Interestingly, previous reports from our group demonstrate a progressive decrease of immature dentine granule cells in Alzheimer's disease patients at breakdown stage 1 and onwards, suggesting that the maturation process of the new dentine granule cells is affected. In this regard, Tobin and colleagues show evidence that the decreased number of immature dentine granule cells in Alzheimer's disease patients is associated with lowest cognitive score, suggesting that the impairments in immature dentine granule cells underlie cognitive dysfunction in these patients. In relation with this data, we also evaluate that the Alzheimer's human tissue at later stages of the illness presents an increased number of cells with 2 or more primary apical dendrites. This morphology has been named V-shape. Okay. Sorry. Yeah. This morphology has been named V-shape. However, today, it was unknown at which stage of the illness the morphology of dentine granule cells is affected, and if there are different vulnerabilities of dentine granule cells according to granule cell layer location. Based on these antecedents, the aims of this project were characterize the morphology of dentine granule cells located in outer and inner regions of the granule cell layer in neurologically healthy controls, determine the morphological alteration in dentine granule cells throughout Alzheimer's disease progression, and evaluate the putative differential vulnerability of dentine granule cells to Alzheimer's disease according to the granule cell layer location. Here, I show you the epidemiological data of the 22 subjects that were included in this study, 5 neurological healthy control subjects, and 17 Alzheimer's disease patients distributed along the 6 neuropathological stages. These experiments were done in collaboration with Dr. Alberto Rábano of the Brain Local Bank of CIEN Foundation. To address the objectives, human hippocampi were dissected and immersed in Golgi solution. The Golgi-Cox staining was performed, and the hippocampal section were obtained using a vibraton. Dentine granule cells were drawn using an inverted optical microscope coupled to a camera lucida, and a total of 639 neurons located either inner or outer regions of the granule cell layer were traced. The cells were reconstructed using the Neuron G plugin for Fiji software, and this reconstruction were used to determine the morphometric analysis that I will detail in the next slide. First, to determine the total dendritic length, all the dendritic tree was measured. For show analysis, concentric rings were drawn and the number of intersections of the dendritic tree in each ring was quantified. The area of the soma was quantified. The number of ending tips were counted manually. Furthermore, dendrite complexity index was determined using the following formula that takes into account several parameters of the dendritic tree. We measure the dendritic span, and we count the number of primary apical dendrites in the cells. And finally, the number of dendrites in each branch order was quantified. In this figure, I show you the representative image of Golgi stain in human hippocampus and magnification of outer and inner dentine granule cells and their respective somas. In the colored arrowhead, we observe the apical primary dendrites in each cell. The quantification of total dendritic length demonstrates that the inner dentine granule cells exhibit shorter dendritic trees and reducer dendritic branching in shell analysis in compared to the outer dentine granule cells. Moreover, the soma area of inner dentine granule cells is smaller and present less number of ending tips. The analysis of the dendritic complexity index revealed that the inner dentine granule cells present less complex dendritic trees and a smaller dendritic span than outer dentine granule cells. Interestingly, a reduced percentage of cells with more than one primary apical dendrite was observed in inner dentine granule cells, which is according with less number of dendrites in the proximal branches without modifications in distal branches between groups. In spite of the fact that the Golgi staining is not selective to differentiate between developmentally born and adult-born dentine granule cells, our data suggests that inner dentine granule cells exhibit a less complex morphology, which is compatible with an immature phenotype similar to that observed in adult-born dentine granule cells. To study the morphological alteration of the dentine granule cells throughout Alzheimer's disease progression, we studied patients with this condition that were distributed along of the 6 neuropathological stages. The subjects were grouped among the following categories. First, the control group, Bragg-Tau stages 1 and 2, which represent the early stages, Bragg-Tau 3 and 4 as intermediate stages, and Bragg-Tau 5 and 6 that represent later stages. The total dentine granule cells, as well as outer and inner dentine granule cells, were analyzed. In this figure, I show you representative image of Golgi stain in human hippocampi and magnification image showing the primary dendrites of dentine granule cells and their somas from the control group and the Alzheimer's disease patients at different Bragg-Tau stages. In total dentine granule cells, the total dendritic length progressively decreased as a Bragg-Tau stages advance, without changes in patients at earlier stages in comparison with the control subjects. Similar alterations were observed in outer dentine granule cells. Meanwhile, the inner dentine granule cells only show a decrease at late stages of the disease. The shell analysis in total dentine granule cells reveal a progressive decreased number of intersections in Alzheimer's disease patients at intermediate and later stages. Similar decrease was observed in outer dentine granule cells population, Meanwhile, in inner dentate granule cells were decreased only in patients at later stages. In relation with the soma area quantification, total dentate granule cells present a reduced soma area at early and later stages of Alzheimer's disease without changes at intermediate stages. Moreover, the reduction in soma area was observed only in outer dentate granule cells at later stages, without changes in this parameter in the inner dentate granule cells. Furthermore, the number of ending tips presents a progressive decrease as the disease advances, reaching statistical significance at intermediate stages. In the case of the outer dentate granule cells, decreased number of ending tips was observed at intermediate and later stages. Meanwhile, inner dentate granule cells are only decreased at later stages. The dendritic complexity index showed a progressive decline in total dentate granule cells, reaching statistical significance at a significant reduction in this parameter. Interestingly, outer dentate granule cells demonstrate a significant reduction of the dendritic complexity index at early stages and onwards. And in the case of the inner dentate granule cells, this decrease was observed at intermediate stages and onwards. In the case of the dendritic span, was only reduced at later stages of the total dentate granule cells. Moreover, this alteration is also observed in inner dentate granule cells at the same stages without changes in the outer dentate granule cells in this parameter. Interestingly, in total dentate granule cells, the percentage of cells with more than one primary apical dendrite was higher in the Alzheimer's disease patients in all Bragg-Tau stages, indicating that this parameter is remarkably altered at the beginning of the disease. Moreover, these alterations was also observed in inner dentate granule cells at early and intermediate stages, and only observed at final stage in the outer dentate granule cells. Furthermore, the number of dendrites in the proximal, the number of cells with dendrites in the proximal branches show an increase at early stages, whereas the number of cells with dendrites, with a decreased number of dendrites in the distal branches is reduced at intermediate and later stages in total dentate granule cells. In the case of outer dentate granule cells, the reduction in the number of cells with dendrites with branches in the distal branches, was observed only at intermediate and later stages. And in the case of the inner dentate granule cells, the number of branches, of proximal branches, was observed at early stages in inner dentate granule cells, and then a decreased number of dendrites in distal branches at intermediate and at later stages were observed. Altogether, these results indicate that total dentate granule cells show early morphological alterations that are further aggravated as the disease progresses. When the total dentate granule cells are analyzed, the first alteration that appear are the reduction in the soma area and the increased number of cells with several primary apical dendrites, as well as the increased number of dendrites in each in the proximal or in the proximal dendrites. Now, as the disease progresses, the majority of the dendritic alterations appears. On the other hand, the analysis of outer and inner dentary granule cells separately demonstrates differential morphological alteration, being the dendritic complexity index firstly affected in outer dentary granule cells at early stages, and then at intermediate and late stages, most of the morphological parameters are affected. In the case of the inner dentary granule cells, the presence of several of cells with several primary apical dendrites are increased, and also the proximal dendrites are increased, and the majority of morphological alterations are affected at later stages. These results suggest the putative differential vulnerability of these two cells, or these two populations, I'm sorry, to a specific pathological mechanisms that are triggered in the Alzheimer's disease brain. As a conclusions, our data reveal that in neurologically healthy control subjects, dentary granule cells present different morphological features according to the granule cell layer location. The several morphological alterations of dentary granule cells are observed in Alzheimer's disease patients at BRCA 1 and 2 stages, possibly before the appearance of the distinct clinical symptoms of this condition. Our data reveal the differential vulnerability of inner and outer dentary granule cells to Alzheimer's disease, and support the notion that the malfunction of the hippocampus underlies the cognitive impairments observed in Alzheimer's patients. Finally, I would like to thank to the Berenice Márquez Lab for all the support in this project, to the postdoctoral scholarships to allow my stay here in Madrid, to the Dr. Alberto Rábano for his collaboration in this work, to the laboratory fundings, and to the CBM. Thank you so much for your attention.
Thank you very much, Berenice. Very nice presentation. So the paper is now open for discussion. So if anyone is interested in posting a question to Berenice, please do so by using the raise hand option in your application, in your computer. Or if you prefer to do so, or you don't have a microphone available, you can send also the questions through the chat, and I will read the questions to Berenice. Okay, so while we wait for the first question, let me just remind everyone that this is the last webinar before the summer break, and we are now trying to close the program for the upcoming webinars after the break from September to December, and we are indeed having 2-3 free slots available. If anyone is interested in presenting his or her work after the summer, please do so by sending us an email either to myself or to Teresa Iglesias, who is the training program coordinator, and I will try to accommodate everyone's interest. So, any questions? I don't see any raised hand yet, or no questions coming through the chat, I believe. Okay, so I'll try to break the ice. See anyone. So Berenice, just a very general questions. So you've quite well characterized morphologically the difference between the granule cells morphology, branching, number of and size of the dendrites and processes. General question is, how do you feel that these differences in the morphology of the cells will affect the function of these particular cells or the circuits of the cells who are connected or these are connecting to? Any idea of that?
Yes, possibly that this alteration, this morphological alteration that we observe in the outer and inner dentine granule cells could be implicated in the, due to the location of these cells, how the afferent connections arrive to them could be stay affected for these impairments in the dendritic tree. So maybe the afferents could be that the last afferents that are arrived to these cells are they are done well. It's not possible to make it due to the impairments in the dendritic tree, or also it's possibly that the morphology of bead shape that is increased in the dentine granule cells could be generate another type of processing in the information that arrives to the dentine granule cell, and could be stay making this underlying the alteration in the information and then in the memory or in the cognitive function. So this morphology affects the processing of the information, not only due to that the afferent connection can be made, and also the change in the morphology can be affected and process malprocessing. Mm-hmm.
So is there any information on whether the inner and outer cells are differentially affected by the pathology, or the amyloid or tau pathology are affected both cells, both cell types in a similar way?
For example, in our lab, in one article from our lab, we observe, and that is one of the antecedents that I put, we observe a decreased number of immature dentine granule cells that are mainly located in the inner region, and the quantification of the total mature dentine granule cells was not affected. So possibly the new dentine granule cells can be affected in a different manner. Mm-hmm. So they made more vulnerable, but at least with the results that I present you today, we observe this differential vulnerability, but just with the morphology and location, we need to make another type of experiments to stay sure about the developmental origin of these cells. But according to our antecedents and these results, we can say that these cells present differential vulnerability.
Okay. All right, thanks.
Yes.
Okay, any more questions from the audience? Okay, I don't see any other raised hands. I don't see any messages coming through the chat. So don't be shy if you have any questions. I guess in any case, if anyone is interested in discussing further with Berenice, I think she will be available to be contacted through email or anything after the webinar. But you still have a chance to make questions if you're interested now. No questions? Okay, so if there are no more questions, I think it's time to close the session. But not before thanking again Berenice for her willingness to share her work with us, for interesting presentation.
You're welcome.
And since this is the last webinar before the summer break, as I said earlier, let me just also thank for the continuous help of Adriana Frontera, Aina Frontera, Celia Lopez Menendez, and Teresa Iglesias in organizing this webinar series. And hopefully we will resume the series after the break in September. I don't remember now which is the first date, but it will be on the website anyway. So thank you very much again, Berenice. Thank you everyone for attending, and I wish you all a great afternoon. And if we don't see before, just have a great summer, everyone. Bye-bye.
Bye. Thank you.